Good morning, children. Welcome back to our online session. In today's session, we will start chapter number 1, Heredity and Evolution. Heredity and hereditary changes. Transcription, translation, and translocation. Evolution. Evidences of evolution. These are the topics we have to be covered today. So now let us move to the first topic, heredity and hereditary changes. One picture is shown. What is the meaning of heredity? The passing of genetic traits from parent to offspring. That is the simple meaning of the word heredity. Transfer of biological characters from one generation to another via genes. That is the definition of heredity. Johann Greger Mendel is pioneer of the modern genetics. He is known as the father of genetics. Research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes, through which 1944 trio of scientists Oswald Avery, Macklin McCarthy and Colin MacLeod proved that except viruses, all living organisms have DNA as genetic material. Only virus is having RNA. Other all organisms are having DNA as their genetic material. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, and in industrial processes in which microbes are used. That is the use of this branch of science. With the help of this science, doctors can find out early detection of some of the genetic disorders. Next is the mechanism of heredity. Transcription, that is the first one. In the picture, RNA polymerase, RNA nucleotide and transcription. These all have shown. With the help of RNA, the genes present in the form of DNA participate in the functioning of cell and thereby control the structure and functioning of the body. Information about protein synthesis is stored in the DNA and synthesis of appropriate proteins as per requirement is necessary for body. These proteins are synthesized by DNA through the RNA. This is called as central dogma. You have to learn the term central dogma, its definition also. The sequence of nucleotides in RNA being produced is always complementary to the DNA strand used for synthesis. Besides, there is uracil in RNA instead of thymine of DNA. This process of mRNA synthesis is called as transcription. In RNA and in DNA, there is a slight change of the amino acids, adenine, guanine, uracil and cytosine. Instead, here it is, instead of thymine, it is uracil. That is the difference. So that you should know. In RNA, uracil is there, but in DNA it is thymine. That's about transcription. Translation, that is the next mechanism. mRNA, messenger RNA, transfer RNA, ribosomes code. Yes, it is coded message from DNA. The message contains the codes for amino acid. The code for each amino acid consists of 3 nucleotides. It is known as, or it is called, triplet codon. Dr. Hargovind Khorana, a scientist of Indian origin, has made an important contribution in discovery of triplet codons for 20 amino acids. For this work, he has been awarded with the Nobel Prize in 1968 along with 2 other scientists. Each mRNA is made up of thousands of triplet codons. mRNA means messenger RNA. As per the message on mRNA, amino acids are supplied by the tRNA or transfer RNA. For this purpose, tRNA has anticodon having complementary sequence to the codon on mRNA. This is called as translation. This in tRNA, anticodon is there. This which all messages mRNA is sending, that will be anticoding and sending to the cells. This process is known as translation. Next mechanism is translocation and mutation. All the amino acid sequence you can see: thymine, adenine, cytosine, guanine, and the replacement happening in the second diagram. The ribosome keeps on moving from one end of mRNA to another end by the distance of one triplet codon. This is called as translocation. Living organisms can produce new individuals like themselves due to genes only, and some of those genes are transmitted to the next generation without any changes. Any nucleotide of the gene changes its position, that causes a minor change, which is nothing but the mutation. In the second diagram, you can see some of the amino acids are replacing, and due to that only this birth time defects. Some children, they are in birth time only they have some defects because of the mutation in their genes. Mutation may cause the genetic disorders like sickle cell anemia. This is an everlasting process, and it is one of the proof for Darwin's theory of natural selection. That is about mutation. Yes, next is evolution. One animal tree is shown in the picture. Look into the picture. Down root is like a unicellular amoeba. Then other one, flagella one, paramecium. Lower type of animals and plants, moss plant and all, you can see in the lower side of the tree. And in the upper side, you can see varieties of big plants and animals. So what is evolution? Evolution is a gradual change occurring in living organisms over a long duration. This is a very slow going process through which development of organisms is achieved. Very clearly it is shown in the diagram. 3.5 billion years ago, life had been non-existent on the earth. There was no life. At present, crores of species of plants and animals with a huge diversity regarding shape and complexity are present on the earth. Animal diversity ranges from the unicellular amoeba and paramecium to man and the giant whale. From the unicellular paramecium or amoeba to the very big animals, dragon, horse, elephant, human beings, squirrel, butterflies, whale, frog, and here peepal tree, corn tree, all different varieties of animals, everything is originated from this unicellular animal. That is the meaning of, that is what is given in the evolution theory. So very clearly it is shown in the picture also. Theory of evolution, Charles Darwin's theory of evolution. In the picture, from the apes to the human beings, the gradual change you can observe from the diagram. According to this theory, the first living materials or protoplasm has been formed in ocean. In due course of time, unicellular organism was formed. Gradually changes occurred in the unicellular organism from which larger and more complex organisms were formed. Duration of those changes is at most 300 crore years. See the time, 300 crore years. Progressive development from plants and animals, from the ancestors having different structural and functional organization, is called evolution. That is another one definition of evolution. That is a progressive development from plants and animals, from the ancestors having different structural and functional organisms. That's the meaning of evolution. Morphological and anatomical evidences. They have to give the evidence or the proof for the theory. So first morphological evidences. Here you can observe different animals, dog, sheep and wolf. And these similarities like structure of mouth and this position of ears, of nostrils and ear pinna are seen in the animals, whereas similarities in characters like leaf shape, leaf finish, etc. occur in plants. It proves that their origin must be same and must have common ancestors. That is the proof of that origin will be same only. Then another one picture also you can see this anatomical evidence, bones, different human bones and cats, then flipper of whales. This from the picture you can observe there are similarities in the structure of bones and bony joints in organs of each of these animals. These similarities indicates that those animals must have common ancestors. That is the evidence. From the evidence they can prove that they have a common ancestor. Vestigial organs and paleontological evidences. That is the next evidence. So what you meant by vestigial organ? Here this human appendix, large intestine, below you can see a small projection, that is the appendix. So what he meant by vestigial? The degenerated or underdeveloped useless organs are called vestigial organs. Example, appendix, which is useless to human, is useful and fully functional organ in ruminants. Various vestigial organs like tailbone, coccyx, wisdom teeth and body hairs present in the body of human beings. Large number of organisms get buried due to disasters like flood, earthquake, volcano, etc. Remnants and impressions of such organisms remains preserved underground. These are called as fossils. Fossils also you can see on the screen there. What do you mean by fossils? Many years back, due to some calamities, whole village will be underground. That time all the animals and plants, here few animals structure you can see, fish, snake, rabbit, crow, these all is buried inside the soil, and after so many years you can see the skeleton of these plants and animals. That is known as fossils. Impressions or remnants. Study of fossils are called paleontology. In science, one separate branch is there. The branch is known as paleontology. Then carbon dating is another one method. Carbon dating method is based upon the radioactive decay of naturally occurring C14 and it is developed by Willard Libby. This method is used to find the age of the fossil. Yes, how many years back, whether it is 200 or 1000 years back the fossils were there. And nowadays this is using for the investigation of any case, any death case and all investigation. They are finding out the age or how many days back this death occurred or the murder happened. That all investigation is also doing with the help of carbon 14 dating method. That's about vestigial and paleontological evidences. Next is connecting and embryological evidences. Connecting links, here peripatus. It is a connecting link between Annelida and Arthropoda. And lungfish, another one. Connecting links. Some plants and animals show some morphological and anatomical which they can be related to two different groups. Hence, they are called as connecting links. Example, in peripatus, characters like segmented body, thin cuticle and parapodia like organs are present. This indicates that peripatus is connecting link between Annelida and Arthropoda. Here you can see the picture of both peripatus and lungfish. Comparative study of embryonic development stages of various vertebrates given in the picture shows that all embryos show extreme similarities during initial stages and those similarities decrease gradually. Here 2-3 pictures of embryos you can observe. The first diagram is embryo in the in the starting, in the growing stage only, beginning stage only. In second, it is little developed. And in the third stage, we can see it is well developed. And each embryo possesses its own structure. Example, fish, salamander, then this chicken, and the one pig, crow, rabbit, man. All in the third stage, we can find out the structure of their parents. But in the first stage, almost all the embryos are having same shape. That is about the embryological evidences. Next, assignment part. Define heredity and explain the mechanism of hereditary changes. Define vestigial organs. Write the names of some vestigial organs in human body. Write the names of those animals in whom some organs are functional. Both the answers are given in the beginning, first and second slides. Definition of hereditary as well as vestigial organs. It is given along with the diagram, the name of the organs, diagram, everything is given. You can copy from the video itself the answers. Explain the theory of evolution and mention the proof supporting it. Theory, evolution theory also point wise are given in the slide. You can write the answers from that. Define fossil and explain importance of fossil as proof of evolution. Yes, in that page or slide you can find the definition and the importance of fossil proof. And one more, complete the following diagram. This is from the textbook exercise which is given. You can complete the evidences, morphological and similarities in bone, and remaining two you have to find out and write. That's about today's video. And thank you. Have a nice day.